Welcome to Folding Feelings, where misfolding isn't just cellular, it's emotional. Let's meet the proteins in this tragic little love story. In the ribosome, a new protein is born. But folding, that's where things get complicated. First up, protein. Fresh out of the ribosome, fresh out of luck. She just wants to fold right, and maybe fall in love along the way. Pero minsan, kahit may sequence ka na, mali pa rin ang pagkaka-fold.
What if I fold wrong? What if I'm useless?
Now here's Helix, perfectly folded, emotionally unavailable. He says he's stable, but his past, not so much. Minsan, yung akala mong functional, may baggage din pala.
Not useless. You just need help. Let me guide you.
With the right bonds, Protein begins to fold. Beta sheets, alpha helices, and feelings.
Why do I feel stable with you?
Because folding is easier when someone sees your potential.
Ouch! Wait, something's not right. I misfolded.
No, don't give up on me.
Oh no, she's...
And this is Chaperone. She's not here to love, she's here to assist. But the truth, di lahat ng tumutulong nananatili.
I'm forgetting. She has to go. It's irreversible. No, I just wanted to fold right.
And sometimes, even with the right guidance, proteins misfold and hearts break.
In another life, we could have folded together.
Hey everyone! We're Group 8 of BSMT 1E, and today we're unfolding, pun intended, the science behind protein folding. So you've heard of proteins, right? No, not the gym bro kind. We mean the ones in your body doing everything behind the scenes. Speeding up reactions, holding your cells together, passing messages, and even fighting infections. But here's the twist. Proteins only work if they're folded into the right 3D shape. If not, it's giving nothing. Actually, it's not functioning at all. It's like handing someone a paper airplane but forgetting to fold it. Not fun. The folding actually happens while the protein's being made inside the ribosome. And it's not random. Forces like hydrogen bonds, hydrophobic interactions, and disulfide bridges are out here guiding the whole thing. And if that process goes wrong, yikes. Misfolded proteins can mess up cells and lead to serious diseases like Alzheimer's and cystic fibrosis. So yeah, protein folding, it's a pretty big deal. Let's get into it. Here are the learning outcomes for today's lesson on protein folding.
Whoa. Do you even know what you're going into? Do you even know what the basic concepts are? Well, not to worry, because I'm here to help. To recap, protein structures are complex molecules made up of amino acids linked by peptide bonds. Their function is determined by their structure, which has 4 levels. The first is the primary structure, in which the linear sequence of amino acids in a polypeptide chain is dictated by the gene encoding the protein and is held together by covalent peptide bonds. The second structure has local folding into structures like alpha helices and beta pleated chains and is stabilized by hydrogen bonds between the backbone atoms. Then, the third tertiary structure is the three-dimensional shape of a single polypeptide and is formed by interactions among side chains, which are R groups, including hydrogen bonds, disulfide bridges, hydrophobic interactions, and ionic bonds. And lastly, quaternary structures is the assembly of 2 or more polypeptide subunits into a functional protein, with hemoglobin as an example. Now, I think you're now ready to tackle the topic itself.
Don't you ever wonder how those different levels of protein structures start to form into themselves? Well, let's go back to the 1950s and 60s, a time when one scientist, Christian Anfinsen, made a groundbreaking discovery that changed how we understood proteins forever. Anfinsen worked with an enzyme called ribonuclease A, and he wanted to answer a very important question. What determines how a protein folds into its functional shape? To explore this, he designed a simple experiment. First, he denatured the enzyme using 2 chemicals, urea, which disrupts non-covalent interactions, and beta-mercaptoethanol, which breaks disulfide bonds. This process unfolded the protein completely, stripping it off of its structure and function. Now, here's where it gets exciting. When he removed the denaturing agents, something remarkable happened. The protein spontaneously refolded into its original active form, without any help. It found its way back, all on its own, to the same shape it had before. This experiment demonstrated a major principle in molecular biology. A protein's primary structure, which is its unique sequence of amino acids, contains all the information necessary to fold into its correct three-dimensional structure. It also showed us that protein folding is a spontaneous and thermodynamically driven process. Anfinson's work was so influential that he was awarded the Nobel Prize in Chemistry in 1972. Thanks to his research, we now understand that the blueprint for protein structure is written directly into the sequence of amino acids itself. Anfinson's experiments told us that all the information needed for proper folding is encoded in the protein's primary sequence. But that leads us to another important question. What exactly drives this folding process? What forces help a protein go from a simple string of amino acids to a complex functional three-dimensional structure? Let's start with the mastermind behind it all, thermodynamics. A protein will naturally fold into the shape that gives it the lowest possible free energy, its most stable form. It's like nature's way of saying, let's keep things efficient and stable. Several factors contribute to this: your short-range interactions, your long-range interactions, and your hydrogen bonds. Your short-range interactions help form your regular structures like your alpha helices and your beta sheets, while your long-range interactions are between amino acids that may be far apart in the sequence itself, but close in space. You also have your hydrogen bonds, which are highly directional and help guide proper orientation. Each of these interactions help the protein become more ordered and energetically efficient. Once enough of them are formed, the protein reaches its lowest free energy, which is its most stable and functional shape. If the protein isn't in its most stable form yet, it continues to adjust, refolding until it finds the perfect balance, the state with minimum energy and maximum function. Now, once your folding begins, certain molecular forces help shape and stabilize the structure. One of the most important are your hydrophobic interactions. One thing that you should know is that proteins usually exist in water, and not all parts of your protein like water equally. Your proteins have hydrophobic side chains, or your water-fearing side chains. These side chains want to get away from the watery environment. So, during folding, these side chains are tucked deep into the protein's core, while the hydrophilic, or your water-loving parts, stay exposed on the surface. This creates a hydrophobic core, which helps stabilize the folded protein, and sometimes even forms pockets or channels that are essential for its function. Now, once your folding begins, certain molecular forces help shape and stabilize the structure. One of the most important are your hydrophobic interactions. There are two major processes or models that describe this process. We have your framework model and your hydrophobic collapse model. In your framework model, folding begins with the formation of local secondary structures such as your alpha helices and your beta sheets, which are built through short-range interactions, as I have mentioned. So, once these stable structures are in place, they gradually come together to form the full three-dimensional shape of the protein, known as your tertiary structure. Your framework model suggests a much more step-by-step process in protein folding process. So, you first collect the pieces and then assemble them together, much like putting together a Lego set. On the other hand, the hydrophobic collapse model starts with rapid inward folding. As soon as the protein chain is exposed to an aqueous environment, the hydrophobic or your water-fearing side chains would quickly collapse toward the center to avoid contact with water. This creates a loosely packed, disordered glob. From this collapsed state, the protein gradually rearranges and fine-tunes itself to form its correct and stable structure. In this model, compared to your framework model, the general shape would come first, and specific structures like your helices and sheets would form afterward. However, it is important to note that both models provide valuable insights into how proteins fold. In reality, many proteins would use a variety or a combination of both pathways to reach their final and functional forms. Another major stabilizing force in your protein folding is a disulfide bond. While many of the forces that hold the protein together are relatively weak and reversible, disulfide bonds stand out because they are covalent. That means they're formed through strong, stable sharing of electrons rather than giving, making them much harder to break. Disulfide bonds are formed when two cysteine residues in a protein undergo oxidation, linking their sulfur atoms together to create a sulfur-sulfur bridge, meaning disulfide. This acts like a molecular staple, locking specific parts of the protein in place and preventing them from shifting out of shape. There are two types of disulfide bonds: stable disulfide bonds, which are permanent and serve as anchors in the final folded structure, especially in proteins that need to function in quite harsh environments, like those found outside the cell. On the other hand, we have our dynamic disulfide bonds, which can switch between oxidized and reduced forms, which allows them to have some flexibility. However, your dynamic disulfide bonds are much less common. So far, we have seen that a protein's amino acid sequence contains all the information it needs to fold, and that thermodynamics and molecular forces guide the process itself. But here's the thing. Inside a living cell, things aren't so simple. The cellular environment is incredibly crowded, chaotic, and full of potential distractions for a folding protein. That's where your chaperones and your enzymes come in. What exactly are your chaperones? Well, your chaperones are special proteins that act like cellular bodyguards. They don't become part of the final protein itself, but they play a critical supporting role. Their job is to make sure that your proteins fold properly without clumping together or folding incorrectly. Some chaperones assist newly made proteins as they're being synthesized, while others step in later during transport or under stress conditions. They don't do the folding themselves, but they create the right conditions for it to happen safely and correctly. In addition to your chaperones, we also have our enzymes. The cell will employ enzymes that actively assist the folding process. These enzymes can speed up specific steps, like forming or reshuffling your disulfide bonds or adjusting the orientation of different peptide bonds to help the protein reach its most stable, functional form. They're the fine-tuners, ensuring that even tricky folds or complex structures are completed efficiently. Together, your chaperones and folding enzymes would provide quality control in the protein folding world. Without their help, many proteins would misfold, lose their function, or even become toxic to the cell. So, while the amino acid sequence might write the blueprint, it's chaperones and enzymes that help build the final working structure, especially in the messy, high-stakes environment of the cell.
So now that we know what protein folding is, let's talk about why proper protein folding is such a big deal. Because trust us, it is. First up, we have functionality. Proteins need a very specific 3D shape to actually do their job. Take enzymes, for example. Their active sites only form correctly when the protein is folded properly. If the fold is wrong, the site won't exist. And without it, the enzyme is basically useless. It's like having a key with no lock to fit into. Second, structural stability. Folding isn't just for looks. It gives the protein its physical and chemical strength. When folded properly, proteins can resist denaturation and degradation under normal conditions. This is important for structural proteins like collagen and keratin, which literally hold your skin, hair, and connective tissues together. Without proper folding, they pretty much fall apart. Third, we have biological specificity. Correctly folded proteins are like puzzle pieces. They fit where they're supposed to. Antibodies need to be perfectly folded to recognize antigens. Same with receptor proteins. They must have the right shape to respond to hormones or neurotransmitters. So if the fold is off, no binding, no signal, and no function. Fourth, we have minimizing cellular stress. When proteins fold incorrectly, they don't just sit quietly. They can clump together and form toxic aggregates that stress the cell out. That's where molecular chaperones come in. They act like folding assistants, making sure proteins fold correctly. And in the endoplasmic reticulum, there's a whole quality control system checking for misfolded proteins. But if these systems fail, you get serious diseases like Alzheimer's, Parkinson's, or cystic fibrosis. And finally, we have efficient cellular processes. For cells to function properly, everything has to work together, like enzymes, transport proteins, and signaling molecules. All of them need to be folded just right to interact correctly. One misfolded protein in the pathway, everything slows down, or even worse, breaks down. So to put it simply, proper folding is what makes proteins functional, stable, and specific. Without it, cells become stressed, systems break down, and diseases develop. So understanding how proteins fold isn't just about passing biochem, it's about understanding what keeps us alive and well.
Alright, thank you so much for that awesome explanation about the importance of protein folding. Now that we know why it's important, it's time for a little plot twist. So what happens when proteins fold the wrong way? Spoiler alert, it's not good. First, let's talk about Alzheimer's disease. Here, misfolded B-amyloid proteins clump together like uninvited guests at a party, except this party is in your brain. Those clumps or plaques mess with memory, thinking, and behavior. Ever walk into a room and forgotten why you went there? Now, imagine that happening all the time. Next, we've got cystic fibrosis. It happens when just one tiny amino acid is missing in the CFTR protein. Just one. It's like making coffee but forgetting the water. The result? Thick, sticky, mucus clogs the lungs and digestive system. Breathing and eating all becomes a challenge. And then there's Parkinson's disease. Misfolded A-synuclein proteins group up inside nerve cells and form something called Lewy bodies. And when that happens, you get tremors, slow movement, and balance problem. Imagine trying to text with shaky hands. Disaster. And lastly, there's Gaucher's disease. In this case, the enzyme glucocerebrosidase misfolds and can't properly break down certain fatty substances in the body. As a result, those fats build up in organs like the liver, spleen, and bone marrow. This causes symptoms like an enlarged liver or spleen, bone pain, and fatigue, showing a single misfolded protein can seriously affect multiple parts of the body. So what's the big lesson here? Protein folding isn't just a nerdy trivia. It literally keeps you alive. When proteins misfold, they can do their jobs, and worse, they might turn toxic and cause life-threatening diseases. And that, my friends, is the consequences of improper folding. So to wrap things up, protein folding is a fundamental process in biology because it decides whether a protein can perform its proper function inside the body. Its specific three-dimensional shape is what gives it the ability to work, stay stable, and interact with other molecules. But when a protein folds the wrong way, it can lose its function and form toxic lumps that damage cells, leading to serious diseases like Alzheimer's disease, cystic fibrosis, and Parkinson's disease, all of which can have life-threatening effects. That's why understanding protein folding is important, as it helps in diagnosing, treating, and preventing these conditions and keeps our cells healthy. And that wraps up our video discussion on protein folding. We are Group 8 of BSMT1E. Thank you for watching, and we hope you learned something valuable today. See you in the next lesson!